Welcome students to my channel BioMinds. So today we are discussing about antibody, the second lecture on antibody, so which includes the function of antibody. So in the previous lecture we have already discussed the structure and the history of the antibody. So if you have not watched the video previous lecture, please watch it first. And today we will discuss certain functions of the antibodies. So there are basically many functions which can be mediated by antibody. For today we are discussing the main four function. So first is the opsonization, next is the activation of complement, next is the ADCC, that is antibody dependent cell mediated cytotoxicity, as transcytosis. So these are the certain functions of the antibody. So we will discuss this one by one. So first we will discuss the opsonization process. So what is opsonization basically? So when there is a phagocytosis process. So what is phagocytosis? Phagocytosis means we have certain phagocytic cells such as macrophages, okay, neutrophils. They act as a phagocytic cells. What they do is they will eat the bacteria or they will eat the pathogen. They will kill the pathogen by engulfing the bacteria inside their body. So that is the process known as a phagocytosis. So what is opsonization then? So opsonization is the process when there is something which promote the phagocytosis. Okay, so when why there is a need of doing this? Because there are certain condition where the sometimes the bacteria, sometimes the antigens are very small amount, so it will be neglected or it will be ignored by the phagocytic cells. So your antibody will help them for the phagocytic cells. So your antibody act as an opsonine which promote the phagocytosis and a process known as a opsonization. So first what it will do, it will first bind to the antibody will first bind to the pathogen. Okay, here you can see this red one is pathogen. These are the antibody. It will bind to the pathogen. And next, the FC portion, the above portion, we have discussed what is FC portion, what is FAB portion. So FC portion of the antibody has, it will bind to the receptors present on the macrophages and neutrophils. Okay, means these are the blue one are the either macrophages or neutrophils because these are the phagocytic cells. So the FC portion, the IgG specifically, IgG molecules, FC portion has affinity towards macrophages and neutrophils. So it will go and bind to the receptor. Here you can see there are receptors present on the neutrophils. It will bind to this FC portion of the antibody and it will eat the bacteria. Okay, so this is how it suppose it promote the phagocytic process. Therefore, the antibody is known as a opsonine and the process is known as a opsonization. That is the promotion of phagocytosis. Okay, so this is the very important function of the antibody. So next from important function is the complement, activation of complement. So what is complement? So complement proteins are the proteins which are present in your blood. Okay, it will help in the immune system. It will help the immune cells or immune organs for the immune responses, as well as this a complement proteins are secreted by livers. There are 30 or 20 more than 20 complement proteins present in our body which helps in the immune responses. Okay, so most of the IgM and most of the IgG subclasses can activate this serum glycoprotein. It is a glycoprotein which is known as a complement. Okay, so the main protein complement protein is C3b. Okay, so it will bind to the antigen antibody complex and it will activate the complement system. So when the complement system is getting activated, it will it will clear the pores on the it will create pores on the targeted cells. So it will create pores on the targeted cells, which is we usually known as a membrane attack complex. So that pores lead to the release of cell contained from the target cells, and the target cells will die. And it also, enhance the phagocytosis. Okay, it will deliver this complexes, that antigen antibody complexes, through liver and spleen for removing, for destroying this targeted cells. So this is what a complement system helps. So here basically antibodies helps in the activation of complement. So how it will do? So IgM and IgG subclasses, so auto IgG subclasses activate your complement system. Okay, because they have the affinity towards this complement proteins. They have the Fc portion has the affinity towards the this complement proteins. Okay, next function is ADCC, that is antibody dependent cell mediated cytotoxicity. So when you see the name antibody mediated cell cytotoxicity, so cell cytotoxicity means which is toxic to the cell. Okay, so here we have the you can say this is the target cells, means if it can be our cells which has antigen in it, or it can be a bacterial cells or the pathogen cells. These are the antibodies. So antibody go and bind to the receptors present on the bacterial cells, and the Fc portions are there. So NK cells, that is natural killer cells, has the CD16 receptor. These are the CD16 receptors, and it will bind the Fc portion of the antibody. So NK cells, this is the NK cells which has CD16 receptors. These are the CD16 receptor. These are the CD16 receptors. This is your bacterial antigen. Uh, your antibody has bound to it. The Fc portion is here. So your CD16, which will get bind to the Fc portion of the antibody. So when they cross link, it will, that NK cells release certain granules, that is degranulation process occurs, and due to the lactic synapses, it will the targeted cells will die by apoptosis. So therefore it is known as an antibody dependent cell cytotoxicity. So it is dependent on the antibody, and cell cytotoxicity means it is cytotoxic to the, it is toxic to the cells. Okay, next function we have is transcytosis. So what is transcytosis? As you all know, we have certain secretions. Okay, we have mucous membrane, which is lining our respiratory tract, gastrourinary tract, urinogenital tract, gastrointestinal tract. So in that mucous membrane, we have certain antibodies. Specifically, we have IgA. Okay, so how does this IgA will be transported from the blood to this mucous membrane? So the process of this delivery of the antibody to the mucosal surfaces, it is known as a transcytosis. Okay, so this is how it will do. Uh, so we have the receptor. Okay, these green one are antibody. It will go and bind to the antibody. Okay, this is blood. Okay, go and bind to the antibody. Next it will do the endocytosis, next the transcytosis. So here it will get released into the other region. So this is how the transcytosis process occurs. So this is all about functions of the antibody. So the first function which we have discussed is uh, complement activation, opsonization, transcytosis, and ADCC. Okay, so this is all about today's lecture. Thank you.